So the next speaker is Chao-Sing Wu from Madison, Wisconsin. I want to make a special announcement. So Chao-Sing is still an undergrad student. She's going to start grad school in Washington soon, but I think she's our kind of youngest speaker of the conference. So without further ado, infer and phylogenetic vectors using algebraic invariants.
Hi. Thank you for having me here. So I'm Chao-Sing Wu. I'm an undergraduate student at Statistical Department in University of Wisconsin-Madison. And I work with Professor Claudia Solis-Lemus in inferring phylogenetic networks using algebraic invariants. So let me first give you some background information. The tree of life is a metaphor and model that is used to explore the evolution of life. It describes the relationships between organisms, both living and extinct. Phylogenetics is the study of reconstructing the evolutionary history of organisms, and it is critical to understanding the evolution. So people like to use phylogenetic tree to understand the tree of life. And here is an example of the phylogenetic tree. As we can see that each node describes an organism and the edges describe the relationships between organisms. So the evolution happens from the root to the top, which is the node of the tree. So there is one limitation with phylogenetic tree is that we cannot represent reticulation in the tree, and it is unable to capture some important biological realities like hybridization, introgression, or horizontal gene transfer. But luckily, phylogenetic network is able to depict this kind of reticulation event. And here is an example of phylogenetic network of wolves and dogs. So phylogenetic network is basically an extension from the phylogenetic tree by the explicit modeling of gene flow. And we can represent phylogenetic network in both rooted and also unrooted version. For the rooted network, here, the red node represents the root of the tree, and we can see that all edges have a direction with it. But in the unrooted phylogenetic network, we are actually suppressing the position of the root of the tree, and also we are suppressing the direction of some of the edges. But we are maintaining the direction of the hybridizing edge, so that in this way, we are actually restricting the position of the root in this unrooted version, as the hybridization edges direction will indicate the time. So there are several existing methods that is used to construct phylogenetic network. The most popular one is probably PhyloNet, which is a pioneer in a likelihood-based method to infer phylogenetic networks. And it provides several alternatives like Bayesian option or like pseudo-likelihood option. And even in like most scalable option, the pseudo-likelihood option, it is unable to handle large number of data or like beyond 100 of genes. And SNAC is a method that is developed by my advisor, Professor Solis-Lemus, and it is available within the package, Julia package PhyloNetworks. And it is an alternative to PhyloNet that it is able to infer network with complexity that does not grow with the number of genes. But still, the method is unable to handle large number of data, like beyond 50 taxa, it will take much longer time. BIS2 is another method that can infer both phylogenetic networks and also gene trees at the same time. And it also can estimate some biological parameters like divergence time. And BIS2 is not suitable for big data because of its complexity problem. So Our goal here is to develop an inference method for phylogenetic network that is scalable enough to handle large data. And we find out that we can achieve this by using something called algebraic invariants. And I'll explain what that is in a few minutes. So our work will focus on estimating networks that involving this type of four-node hybridization cycle. And here's an example of this type of unrooted phylogenetic network. And we can see that the blue edges here are the hybridization edge, and the blue node is a hybridization node. E here represents the branch length of the network, and gamma is the inheritance probability. So it represents the proportion of gene that is inherited. And 0, 1, 2, and 3 represent the clades in the network. So we call this type of network a diamond structure. And here are some examples of this type of network involving the four-node hybridization cycle. So for the network with 8 taxa, one possibility is that we have 2 taxa in each clade. We call this network N2222 because we have 2 taxa in N0, N1, N2, and N3. For this one, this network involving 7 taxa, we have 2 taxa in N0, 1, and 2, and only 1 taxa in N3. So we call this N2221. And similarly, here's an example of N2211 and N1112. And previous work has shown that the distribution of gene trees will tell us about the phylogenetic network. And we also find out that the split frequency of the subset of 4 taxa can also tell us about phylogenetic networks. And we call this split frequency of this 4 taxa subset is a concordance factor. And so concordance factor will be used in our method as a input data. And before I explain what is concordance factor, I want to emphasize that our method can also take in other input data like DNA sequence and gene trees. So we can estimate gene trees from DNA sequences using methods like IQ tree. And the gene trees, we can use it to get the concordance factor table using the Julia package phylo-networks. So our method is able to use DNA sequence and gene trees as input, and we will convert it into concordance factor for our method. So what exactly is concordance factor? As I mentioned earlier, it is the split frequency of the 4 taxon subset. So imagine we have a 4 taxon subset A, B, C, and D. There are 3 possible splits. So it can be A, B, and C, D, or A, C, and B, D, or A, D, and B, C. We call this splits a quartet. And the split frequency or the quartet is basically a concordance factor is basically the proportion of gene trees that is displaying this type of quartet. And we can write concordance factor as functions that is related to the branch length in this way. And if we know what is the value for our branch length, we can calculate the concordance factor value. So our method will use algebraic invariants, which can be inferred from our concordance factor polynomials. So for example, if we have a network, specific network with a diamond structure N, and then we can sample all the possible 4 taxon subsets from it. For each subset, we will get 3 quartets and its corresponding concordance factor polynomials. And we can find out the set of all concordance factor polynomials for this one network N. What we do is that we will assign a variable AI to each concordance factor polynomial. And then the algebraic invariance is basically the relationship that AI needs to satisfy for the concordance factor polynomials to be consistent. So it might be confusing to like what is algebraic invariance, but let me give you an example. So here, for the three concordance factor polynomials here, we can see that they all sum up to 1. Like it's not hard to see. So we will write this relationship as A1 plus A2 plus A3 is equal to 1. And we can get an invariant that is A1 plus A2 plus A3 minus 1 is equal to 0. So the set of concordance factor polynomials is actually going to define a set of invariants that is going to vanish to 0 if the data is coming from the true network that we have. And I want to emphasize that it's not always so easy to like eyeball what is the concordance factors, what is the invariance for our concordance factor polynomials. Sometimes it can get really complicated like this. So, so I can't eyeball this, otherwise I'll be a genius. So we will use a software that is called Macaulay to find out of this invariance. So as I mentioned earlier, like if the data is coming from a network, and then we have the algebraic invariance for that specific network, the algebraic invariance will eventually vanish to 0. So what we do is that if we want to estimate where the data is coming from, we will need to have several candidate networks, and then we will get all sets of algebraic invariants for it. So we will input our data to it and then see which invariant is going to best match with our data. So the question is, how should we find the candidate networks? Do we need to like search the entire network space? But it turns out that there is a paper have proven for this type of diamond structure, if there are, we can identify all possible concordant factor polynomials if there are at most 2 taxa in each clade. And that means that for the 4-node hybridization cycle, we only need 2 nodes in each clade to define all the concordance factor polynomials. So for the candidate network, we can restrict our search for the entire network space to only networks with 5 to 8 taxa. And here are the list of all the candidate networks. So I have explained what is algebraic invariance. Let me introduce the procedure of our method. So if there are less than or equal to 8 taxa for the input data, our method will basically just iterate through all the candidate networks and then calculate what's the invariance score for each candidate network. The network with the smallest invariance score is going to be the network that best matches with our data. If there are more than 8 taxa, what we'll do is that we will get the sub network for our data. So we will iterate through all the candidate subnetwork with only 8 taxa, and then we will also calculate the invariance score. We will set the template as the subnetwork with the smallest invariance score, and then we will add the remaining taxa accordingly to our template network, and so that we can get our eventual phylogenetic network. So our method is just use algebraic invariance, and it doesn't need to like search the network space or do optimization like other most methods do. So it will be relatively faster than other methods. But we also want to prove this by some simulation studies. So the first simulation we're doing is that we are inputting different concordance vector tables. So this is using the true concordance vector table, and then we add different levels of noise to it. The x-axis are different network structure, and each bar is representing the proportion of times that we are selecting either the true network or the symmetric network. So a symmetric network means that so it's a network with 4-node hybridization, and 2 of the clades are actually inverted. That's what we call a symmetric network. It is very close to the true network, but only the 2 clades are inverted. As we can see that most of the time, it is able to select the network that is either true or symmetric. And the second scenario is we are inputting gene trees to our method, and we are evaluating it on 100 gene trees, 1,000 gene trees, and 10,000 gene trees. And as we can see that with larger number of gene trees we feed into our data, we give it more information, and it is easier this way for our method to estimate the correct network. And as we can see that its performance is pretty good. At most of the time, it is able to find out the true network. The third scenario is that we are input the DNA sequence. So the first one is DNA sequence of length 500 that is estimated from 1,000 gene trees or from 10,000 gene trees. And we are evaluating DNA sequence of length 2,000 from 1,000 gene trees and 10,000 gene trees. And as we can see that our method performs pretty good with high accuracy. And we are also interested to compare our method in terms of time with other methods. So we are comparing it with SNAC, which is a method developed by Professor Solis Lemus, and phylonet with the option maximum likelihood and maximum pseudo likelihood. And as we can see that our method is fastest among all and is like more than 10 times faster than SNAC. And after all this simulation scenario, we are interested in how our method performs for real data. So we use the real data that comes from Canis genus, and we are interested to see if our method is also faster in this case. So for this data set, there are only 6 taxa, but our method is like around also more than 10 times faster than SNAC, and also much faster than phylonet maximum likelihood or maximum pseudo likelihood. So we want to know if our method is able to select the correct network. And it turns out that our method is able to find out the correct network that is also shown in the original paper, and also the same network as SNAC and phylonet with maximum likelihood. But phylonet maximum pseudo likelihood actually selected a different type of network here. So to sum up, our method will use algebraic invariance, which can be optimization free, and it doesn't need to search the network space. And simulation study have shown that our method is relatively faster than other method and can perform with good accuracy. And we want to emphasize that our method is able to allow biologists to use as a reference when comparing with other methods, and also allow biologists to like narrow down their search of the network space. So our work is currently available in arXiv, and our method is implemented in this Julia package phylodiamond. I would like to thank my advisor, Professor Solis Lemus, for coming up this method. And I want to thank all my lab members for their help and support. And also want to thank this little buddy over here for his moral support. So thank you. And I would like to take any questions if you have.
for a great talk and keeping time. We have time for questions.
Hi, very cool talk. I was curious, since you use these algebraic invariants, there can be like different combination of values that would lead to the same invariant value. So instead of 1 minus 2/3, 1/3, 1/3 in your example, you could have like 1, 0, 0, and that would still lead to the same inference estimate, but maybe not the same network would, like different networks could give the same invariant value.
So, so for a network, so for concordance vector, it is only related to the branch length of the network, which is denoted as a variable t here. And for the invariants, like this, they are about a variable ai, but then ai are also related to branch length t. So they are the same for the same structure, but then it will change as the branch length change. Does that answer your question?
Yeah. Thank you.
Hi.
Hi.
Oh. Hi, this is really cool. I was wondering if you could provide some intuition as to why algebraic invariants make this computation so much faster.
So for other methods, I not entirely familiar with their algorithm, but they will most of them will need to search the network space and then use optimization to like find out what's the local minimum or local maximum. But our method will only evaluate some polynomial equations, and in terms of time complexity, it is much faster than the algorithm that needs to search the network space. Because when you are evaluating the polynomials equations, you basically need to plug in those values, and the time complexity is mostly just one. So it's much faster that way.
Hi, really nice work. I have two questions. One, can you also use this to estimate the branch length, the parameters of the tree?
Sorry, no, that is actually one limitation of our method. We are only able to identify what are the taxa in each clade, but we are unable to identify the branch length.
And another question, what happens if there is more than one reticulation event, one hybridization event?
So our method right now is only based on one hybridization cycle, and we want to like do that in the future if we can extend our work to more reticulation events.
Right, because I think in the Canis phylogeny they have more than one, right? Or
yeah, exactly. Yeah, I think that's a great future direction.
Thanks.
Great talk. I thought this is as if a PhD thesis disciplines like this level of intelligence. Yeah, great. I have two questions. One is how. So you have a core four node hybridization cycle. How do you determine which branch is the which direction is the hybridization cycle? The hybridization, because I have tried those things, but from the if using CF, I think all the directions the hybridization give you the same CF. So how do you determine which one is the correct CF? And especially when there are ghost the extinct species, and those extinct species you are not sampled like ghost will have a hybridization will have impact on your other species which you are sampled and have in hybridization. I don't know if that's clear.
Yeah, that's a great question. So that is actually another limitation of our method. So as I mentioned in our simulation scenario, we're identifying the true network and the symmetric network. So what I mean is for the symmetric network is that N1 and N2 are actually inverted instead of we know, oh, what is N1 and what is N2. This happens because our method is unable to estimate what is the inheritance probability. So as you can see, if we want to identify what is I1 and what is N1 and N2, we need to identify what is the minor edge and what is the major edge here. And we are unable to do that. So our method is actually not good at identifying between the true and symmetric one.
Oh yeah. I want to make yeah, maybe another argument or like sorry, another question or making it more clear. So I'm not when we are saying if gamma is like for example a half, but can you determine just from this unrooted structure which node among n1, n2, n3, or n4, which one is the hybridized node? Just from ZF or yes. Okay,
Yeah,
can you
that's a great question.
Mhm.
I have to say I really don't know the answer, but yeah, that's something I never think of, but I will think about, and we can talk after this talk.
Thanks very much.
Thank you.
Are there more questions? If not, we're going to have a 15-minute break. Thanks again to all the speakers of the session. And there should be coffee outside.